I do want to introduce our speakers today. So the first session is the natural history of clinical trials. This is Melissa Gamble. She's the president and co-founder of the Global Foundation for Paroxysmal Disorders, and Dr. Mousumi Bose, the medical and scientific research liaison for the foundation. So I'm going to let them do their thing, and as soon as they're done, we'll break for some questions. You know, we'd like to make this as informal as possible. This really is a session for you to be part of. So, you know, please feel free to interact, engage, yell out at them, and then afterwards, if we have a few minutes for questions, then we'll break for lunch. So, thanks.
Thanks. Thank you, Kent. For that great introduction. We're going to piggyback on a lot of what has already been mentioned this morning about the importance of natural history studies. And we hope that after this session, you'll really be energized to go back and perhaps undertake a natural history study if you've not already done that. And we really hope that you see the value of strategically investing in natural history studies. So, who we are. I am the mother of a child with a rare disorder, as is Mousumi. And we are on the board of directors at the Global Foundation for Paroxysmal Disorders. And just to give you an overview of what we're going to be talking about today, we'll give you some natural history study basics, share a little bit about our experience with natural history studies, and just present you with some possible resources for engaging in a natural history study. So there's our new logo, which we launched yesterday, actually, in anticipation of this meeting. And the GFPD was founded in October of 2010. We are a 501c3 nonprofit public charity. We have a 7-member board of directors that are all parent advocates, parents of children with paroxysmal diseases, and we have an 8-member medical and scientific advisory board. And we'll talk later in this session about the importance of the scientific advisory board and their role in natural history studies. One thing to note, our registry contains 383 children worldwide with our disorder. We have 157 living and 226 deceased children and adults on our registry from 30 different countries.
It's actually 31, right? No.
Yeah, I think it is 31 countries. So there's our social stuff, where we also launched Instagram yesterday, which we're really excited about. So, so basically, the GFPD aims to fund and promote research. We organize family and scientific conferences for families and scientific professionals to gather together and collaborate. We host online parent support groups, an equipment exchange program, and we engage in patient and physician education and advocacy. So one of the main ways that we've done that recently is the publication of a set of treatment guidelines, which we're very proud of. Dr. Bose was an author on that document. And that document is really the first of its kind that's taken some of this natural history data and made recommendations for treatment and management of our disorder. And we're advocates because of our kids. So I mentioned this at the beginning. My daughter, Jenny, is the cute little gal with the glasses, and she was born with a paroxysmal disorder. She lived to be 6.5 and passed away last year from the effects of the disease. So she was just an amazing kid and the love of my life. I'm sorry to my husband. But you know, she fundamentally changed the course of my life, as I'm sure many of you can relate to. So I'll let Mousumi tell a little bit about Elan, the cute little guy with the hat.
Yes. So that's my little guy, Elan. He was born in August of 2010. He had a severe form of this disorder, so he was given about 6 months to live. He managed to fight bravely through past his first birthday, so I'm really glad that he we were able to celebrate his birthday. He passed away in October of 2011, and since then I've been trying to honor his memory, sort of keeping his legacy alive through my involvement with the GFPD. So I want to get started with our discussion on natural history studies. The reason I'm showing this slide is to help empower all of us to think about, if you're considering taking on a natural history study and you're a little bit concerned about whether or not you have the tools or the resources to initiate or execute one, I just want to take a step back and remind us that we've already done so much as rare disease advocates. All of these things are done, although they were executed by legislators and by scientific researchers. All of these things were done through the grassroots efforts of patient advocates like us. So we have the ability to do anything, and that includes doing natural history studies. So I will talk a little bit about what the definition of natural history is, and I'm going to have to look at my notes here because it's a long paragraph. So in most definitions of natural history, the natural history of a disease is the natural course of a disease from the time immediately prior to its inception. And over here, exposure can be interchanged for inception, progressing through its pre-symptomatic phase and the different clinical stages to the point to where it has ended and the patient is either cured, is chronically disabled due to the effects of the disease, or has passed away due to the disease without external intervention. So that's the traditional definition of a natural history study. The one thing I want to highlight is that when you're talking about rare diseases, and 80% of these rare diseases have a genetic basis, when we use the word exposure or inception, we're talking about when that mutation occurs. So that exposure, that inception, it happens in utero. So what does a natural history study tell us? Bottom line, it gives us information it should give us information to effectively characterize a disease population, give us a better idea of what that population is like. And this can be done, traditionally natural history studies have been executed with a sort of clinician-initiated effort where clinical measurements were collected by a physician and measured over the course of time. But what's actually become a growing method towards collecting natural history data is through patient-reported outcomes and the use of registries to collect this natural history data. So I'm going to talk a little bit about using both of these resources to fully capture as much natural history data as you can collect. Thank you. Mm-hmm. So why is this important? Why do we care about this? We're here as either parents or as people with rare diseases where there's very limited treatment options, and the first place you want to go is, why do we want to take this on? We need a cure. We need it now. We need to focus on treatments. We need to focus on therapies. The reason for taking on a natural history study, as daunting and as long as it may seem, is because we need this information in order to get effective therapies, to get effective treatments. Mm-hmm. And that's really that's the basis. This is the foundation for developing these well-informed clinical trials, and this will help us improve diagnosis, at the very least get a better estimate of the incidence rate of our diseases. And ultimately, it will help improve the prognosis of the disease as we learn more information about it and we get information about better treatment options. Mm-hmm.
And I want to say something here. Mousumi mentioned that a cure treatment, really that is our focal point, but you can't have that without the foundation of natural history, and that really holds back our ability to find other treatments.
Absolutely.
And it's not the flashiest thing to do, but it's really the foundation. It's important.
And you need that foundation.
Yep.
So one of the things that I want to point out here is that with diseases that have a high mortality rate, it's pretty well accepted, although traditionally a natural course of a disease is looking at how it progresses without external intervention. But natural history studies for rare diseases that have these high mortality rates can also be used to assess the effectiveness of already established therapies, supportive therapies for these diseases. Okay, so now that we've talked a little bit of why these are important, let's talk about how to go about designing a natural history study. So I like to use this quote, begin with the end in mind, and I've heard other people use this quote when discussing natural history studies. And you know, I'm going to go back for 1 second. We talked a little bit about why this matters, having a comprehensive understanding of the disease, improving diagnosis, et cetera. And honestly, these are all relatively broad goals. When you decide to take on a natural history study, you have to go into it with a very specific idea of what you want to do with this information, or that's really the best way to efficiently collect data. So what's really important to do is to think about what's your end game here? What type of treatment options are you looking for? Are you looking at gene therapy to improve CNS symptoms? Then that's how you need to frame your natural history study and experimental design. These are the outcomes that you need to be looking at. So this is actually, I guess I'm sort of saying this because this is a lesson that we have learned in the collection of our natural history data. It can get with PBD, with Zellweger spectrum disorder, it's a multi-organ disease, and there are so many symptoms and so many organs that are affected by, and so it's very easy to get wrapped up of collecting information about this, that, and this, and it gets so daunting. And so having that sort of end game in mind really will help streamline your efforts to initiate a natural history study. So I want to talk about 3 major approaches to collect natural history data, and these can be done independently, but truthfully, the ideal natural history study should incorporate elements of all of these things that I described here. The medical chart review, doing a cross-sectional study, and then ultimately getting to prospectively, longitudinally collecting data. And that's really the gold standard of collecting natural history data, is being able to follow time points throughout the course of a patient's life. So again, this can be either clinician-initiated, or this collection of this data can be clinician-initiated, or it can be patient-initiated. But really, in this day and age, I think the best way to approach it is to have a collaboration between both the clinician, the researchers, and the patients, because that's really going to give you the most comprehensive, the most useful information about what the disease is about and what needs to be treated. Yeah. So I want to just talk, now that we've talked a little bit about experimental design, I just want to put some of these into a framework of an example. So we've already heard about Duchenne muscular dystrophy, and it's because it's been in the news a lot lately about different treatments. And the reason that we are hearing about all this stuff in the news about about treatment options for Duchenne is because over the last several years, there have been a multitude of publications discussing the natural history of Duchenne's. And so this is just a little history of the study itself, what its goals were, to study the spectrum of disease severity, looking at disease progression based on exposure to the available treatments. And what I want to highlight here is that with their outcome measurements, they incorporated all of these elements of these approaches that I just described. They have a medical chart review. They used clinical exam. The 6-minute walk test was actually developed throughout the course of the study as a primary outcome measurement because it gave valuable information on the course of the disease. So they set these things in place. And actually, I want to just break for 1 second. And I want to say is when I talk about the beginning with the end in mind and having these endpoints set up, it's good to have this sort of structure set forward. But it's also really important to have some level of fluidity or dynamicness towards what you are going to measure. And that's actually something that happened in the collection of the Duchenne natural history data, is the discovery of this 6-minute walk test being a very valuable outcome, and then it eventually became a primary outcome for this study. So, sorry, I just sort of broke there for a second. And with the conducting of this natural history study, there's been a number of publications that I've just mentioned, and here's just a few of them. And there has been more information revealed on treatment options for this disease. So one more thing I want to say about the Duchenne natural history study is that there was recently a paper that I don't have listed here that showed they had a patient registry. And here, you look up here, they are looking at quality of life, satisfaction, but also looking at medical outcomes. And what they found is the Duchenne Connect patient registry actually was very useful with reporting medical outcomes of the disease, and actually correlated back to some of the measurements that were taken clinically. So the conclusion of this one paper was that using this registry information can be very, very valuable to the collection of natural history data. Mm-hmm. So I want to just start now and talk a little bit about the GFPD's experience with natural history data collection. And we are nowhere near where the Duchenne muscular dystrophy study is, but we're on our way.
We're getting there.
So we have had the great fortune of having a number of resources available to us to help collect natural history data. So we have, starting off with one of our scientific advisory board members, Dr. Nancy Braverman, who registered on clinicaltrials.gov a study called the Longitudinal Natural History Study of Patients with PBD. And this was registered in 2012. And her goals were to define the PBD population clinically, biochemically, and genetically. And she was also incorporating multiple approaches to collecting this information, looking at medical evaluations and medical charts, and hoping to incorporate this information to informing a prospective longitudinal data collection. And this is actually where I feel like we've really learned a lot from this process, because currently we are still analyzing medical records 4 years later. And the work that Dr. Braverman's group has been just invaluable. They've collected so much information. But now we're sort of at this place where we're talking about moving to the next step, collecting prospective data. And it's been a little bit of a challenge. So,
it has, yeah.
So what we have decided to do is try to initiate this collection of prospective longitudinal data in conjunction with the NORD, the National Organization for Rare Diseases Natural History Project, which happened this past year. So we have set up a registry with the National Organization for Rare Disorders, called the Peroxisome Biogenesis and Related Disorders Registry. And this is with the advisement of Dr. Braverman, who's been helping us through this process. So just a little history on this project. In 2015, the FDA gave NORD $250,000 to fund the development of natural history studies for rare diseases. And what NORD did is they chose 20 patient advocacy groups to use their platform, which they had advertised to be their registry platform, which they had advertised can be used to collect natural history data. And over the last year, we've been trying to initiate this project, execute this project through developing our surveys for collection of this data, and also going through institutional review board approval, and then finally launching the website for where our registry will be housed. So there were 20 groups chosen, including the GFPD. Again, this is a registry to collect patient-reported data, but there are plans of NORD. We have discussed the possibility of including a clinician portal so that clinical measurements can be included in this data collection. So we are planning to launch our registry sometime this year. So before December, we will have our registry launched. And these are the 20 groups that have participated in this project, which are sort of serving as kind of like beta testers for using this platform. And I think as we continue to get more feedback and give NORD more feedback, it's going to be a really efficient way to be able to collect this data.
And one other thing, this data will be de-identified, and NORD can, we own the data, but NORD also has the ability to use the data as well, just to track things across rare diseases.
Right, and they're looking at issues like quality of life, and I think they were looking at like nutrition as well. Yep. Okay, so that's what we'll say about NORD. Another resource that we've recently been, we've had the pleasure of getting involved in is the Sterol and Isoprenoid Research Consortium, otherwise known as STARE. So Zellweger spectrum disorder, peroxisome biogenesis disorder, is a disease of sterol metabolism. So that's how we sort of got pulled into this particular research consortium through the activities of Dr. William Rizzo at the University of Nebraska. And what the STARE Consortium is an affiliate of the Rare Disease Clinical Research Network. And this was briefly mentioned this morning as well. And I'll talk a little bit about the RDCRN in more detail in just a second, but the RDCRN houses 22 research consortia of a number of different groups of diseases. Mm-hmm. And I feel like this is a really good resource for patient advocates and patient advocacy groups to be familiar with. And what it basically is a collaborative effort between both patients and researchers to promote research in these specific diseases. So this is done through the use of a communication registry. And in the STARE Consortium, there are multiple patient advocacy groups that are involved. And we're actually going to have our patient advocacy group meeting in Omaha in just a few weeks. So what we are planning to do with using the STARE resource is that they have a number of clinical protocols that have been initiated by the researchers affiliated with this consortium. And already there is one approved protocol to collect natural history study on SLS. And we hope for our disease that we can also collect some natural history data through this, our affiliation with this consortium. So all of these research consortium, including STARE, they have what's referred to as a contact registry. And it's basically this online communication platform for these patient groups to be informed of clinical trials that may be related to their disease. But there's also a survey platform that's associated with it to collect patient-reported data. And we have plans to actually collect data on quality of life, and nutrition and other elements over the next year or so. So this is something that we very recently come in contact with. So we're still in the very early stages of using this resource for collection of natural history data. So to give you an idea of what we hope to gain from all of these resources that we've had the fortune to be in contact with, Zellweger spectrum disorder, being a spectrum disorder, it's a very heterogeneous disorder, and we hope to get a better understanding of it, a better characterization of our disorder. And perhaps through that develop validated biomarkers related to clinical progression that can be used as targets for treatment options. This is our broad goal. And then and overall have these effects improve the quality of life. Now, my background is in nutrition, so I'm always interested in how nutrition affects rare diseases. So that's also something that we're hoping to undertake. So I'm going to start talking about some resources that may be useful for other patient advocacy groups. And I just want to start by talking a little bit about having an engaged patient population. And we are so lucky, the GFPD is so lucky to have an active active patient community, and this is a bunch of us, not even close to how many are in our group at a fundraiser that we had in Maryland not too long ago. And so having that patient involvement is crucial to collection of natural history data, because that's where you're getting your information from. So making sure that your community is involved, is excited about collecting this information, because that's what's going to help with treatments, is really, really important. And it's also really important to have an engaged research community, and we are also very fortunate that we have a very active research community. We've got a scientific advisory board of how many members? Like 7? 8 members, and they are all so passionate about helping our kids, and we just feel so lucky to have them be a part of our community. So in terms of some tools to initiate a natural history study, I mentioned the engaged patient community and research group. So do we have some patient advocacy groups that have scientific advisory boards? Good, fantastic. They are so useful in helping develop studies like this, and there's no way we could have taken on these efforts without their involvement. So in addition to having these groups, you have to have the ability to collaborate, whether this be through face-to-face meetings, whether this be through conference calls, or whatever. And another thing that you really need is a means to collect and organize data. So we are hoping to do this through our NORD platform and through our STARE online survey platform. And here is the website to find out more. If you're interested in learning more about developing a NORD registry to collect natural history data, this is the website, and there are some people that I have acknowledged at the end that would be good contacts to learn more about this particular resource. And then finally, a really important aspect of a natural history study that you have to have is the ability to request institutional review board approval and HIPAA certification. And this is really where having a scientific advisor or medical advisor who's affiliated with an institution that has an institutional review board really is helpful. Now, through NORD, they're actually using an external IRB service. So it's not absolutely mandatory to be affiliated with an institution nowadays. So that's also another tool.
Yep. And I want to hop in real fast and talk about, in terms of collecting and organizing data, that's one of the real issues that we've had with the longitudinal natural history study that Dr. Braverman, not an issue, but a challenge that we've had is collecting and organizing this data, because she's looking for clinical information, she's looking for biochemical and genetic information, and it's just so much data.
Such a daunting task. I think Dr. Braverman has had their clinical coordinators, they don't do anything but try to collect this data, and I feel sorry for them, because it's such a daunting task.
It is. So having a means to collect and organize it is just crucial.
So I mentioned that I was going to talk about the RDCRN again. So what the RDCRN is, it's basically a product of the Rare Diseases Act of 2002. Okay, and this was one of the things that came out of it. And it was basically a collaboration between NIH, researchers, and patient advocacy groups to collaborate on information regarding treatment of rare diseases and clinical studies for the disease, and to raise awareness for the disease. And one of the mandated tenets of the RDCRN, in order for it to be refunded, is that there has to be active engagement of the patient community. So being involved in the RDCRN I feel like has been really useful for us, but it's actually extremely useful for the researchers as well, because it's mandatory. And I knew nothing about the RDCRN until this year, when Dr. Rizzo had approached me and said, do you want to do you want your group to be involved in this particular consortium? I was like, how did I not know that? So I don't know. I'm sure there are people in our audience that are much, much informed, more informed than I am. But if you are not familiar with the Rare Disease Clinical Research Network, you should definitely see if there is a consortium that may serve your particular disease. There are, as I said, there are 22 research consortium, and they encompass a broad spectrum of rare diseases. They've got a brittle bone disorders, they've got a mitochondrial disorder. So it's do I have the website here? Yes, there is the website. And what I want to sort of point, I was talking to some of the PIs of our consortium not too long ago, and one of the things, and I was telling them that we were going to do this presentation, and one of the things they wanted us to highlight is that not only is it informative in terms of getting information about clinical trials, perhaps collecting longitudinal data, it's even useful in terms of bringing patients together that are not originally affiliated with a patient advocacy group. They said that there was one of the diseases that they were looking, there was no patient advocacy group until they started reaching out to these patients and bringing them into that specific consortia. And now, as a result, there is a patient advocacy group that's been formed. So the RDCRN has the ability to do so many things in terms of using this collaboration between researchers and the patient community. So finally, the last thing I will say is this is just something that sort of popped up on our radar a little while back. In May, the FDA announced this grant opportunity to fund targeted natural history studies for rare diseases. And these funding opportunities are pretty, I think, speaking as a patient advocacy representative, this is a lot of money. The RFA is here, and it says that any entity can apply for this. It's really helpful and useful to bring a scientific advisor into efforts like this, but they are actually having 2 cycles. One actually ends in about a month, so I wouldn't recommend that you try to make that deadline. But there is another one that is the deadline is 2018. So this is a feasible goal that you can where you can actually get funding for developing your natural history study. Okay, so again, here is the RFA for that. Please feel free to look into it. So I want to finish by just saying that the lack of natural history data on rare diseases is the most common issue that hinders the development of effective treatments for rare diseases. And I'm not just saying that because I think that. The FDA thinks that. I'm assuming that there are pharmaceutical industries that think that too. And in order to collect natural history data, you cannot do that without an active and engaged patient community. But that being said, through the current resources that we have available, and what I want, something I wanted to say that I didn't say before is that I've described a few resources here, but I'm guessing that there are members of the audience that have access to other resources that we don't even know about, and I'm hoping that we'll have time to discuss that. Using the resources that we have available now, the patient community can play a major role in not only participating in these studies, but also really shaping and molding where this research goes to provide the most beneficial and useful information to serve that patient community's needs. Mm-hmm. So I want to thank our coordinators for our longitudinal natural history study, Dr. Nancy Braverman, and these are her 3 clinical coordinators. I want to thank NORD, and these are some of the key people to contact if you are interested in looking into using that platform for collection of natural history data. And then I wanted to thank the PIs of the STARE Consortium, Dr. William Rizzo, Dr. Jean-Baptiste Roulette at Washington State University. And I want to thank Global Genes for having us here today, and I want to thank PRA Health Sciences for sponsoring this session. And here's more information about our organization. Thank you very much.
So thanks to both of you. That was great. I think we're going to open it up for questions and see if you have any interaction you want to have with these folks who clearly know what they're talking about. I think I have spent my career in clinical drug development and clinical research, and it is one of the fundamental problems is not understanding kind of the natural history of the disease before we figure out how to fix it.
Right.
And so you talked a lot about chart reviews and the challenges of trying to get all that data together. I mean, one of the key things that I focus on is really leveraging the world of electronic healthcare data and mining that data through artificial intelligence and natural language processing. And I'm just wondering, on those chart reviews you're doing, they're so manual. Do you have anybody that's working on kind of electronic health record reviews and doing that through data analytics?
That's a really good question. So the clinical coordinators in Dr. Braverman's group, they're just fantastic people, and they're very enterprising. So one of the clinical coordinators has come up with this kind of, her goal is to develop an app for this in terms of collecting this type of information. But in terms of going back to the data that's already there and sort of mining through it, right now it's just been extremely manual and very, very daunting. But I think that's definitely going to be the future of collecting this type of information. We just don't. We're still trying to work out the kinks. Did you have something that you wanted to add?
No, I agree. I mean, we're moving that direction, I think. So.
I mean, it's hard. And I think most patients and patient advocates know the healthcare system is not moving as quickly as the rest of the world when it comes to technology. And so we're trying to get there very slowly. But
I think standardization of language is going to be really key. And I mean, I know that there is already guidelines set in place for standardization of language when writing down medical records, but it's still, they're guidelines, and so sometimes going through these medical records it's like there are two medical records that are describing the same thing, but it's completely, you can't compare the two because they're written in completely different languages. At least that's something that some of our clinical coordinators have sort of, lamented to us about.
Well, and I think one of the problems with the community and the physicians is that, for most of these diseases, we don't even really have ICD-9 or ICD-10 codes.
Right.
So physicians don't even know how to put it in the chart. So when we're trying to find natural history information, it's more of a detective story than say, if it was lung cancer or heart disease.
Right.
So.
Absolutely.
Are there any questions in the audience? So the woman in the black and white right there.
Hi.
My daughter was born with an extremely rare disease. She's one of 7 living in the world so far that have been diagnosed. And thankfully, a dear friend of ours who's an RN told me about 6 months in when we started discovering there was more and more issues. We actually weren't even diagnosed genetically until a year ago.
Wow.
She celebrates her 9th birthday next month.
Yeah.
But we had all these other presentations, and many people had said because my pregnancy was normal, everything was fine, that they said, I really think there's just something genetic going on. I started at 6 months a natural history where I listed from that point on to currently every med we've ever tried, her interactions with it, every type of equipment, every genetic diagnosis,
fantastic.
That's awesome.
every lab test, you name it. My question to you is, and I don't know if it might be on those specific websites that you listed, it's now 16 pages. And what I do is, besides listing those labs and things, I journal every single night with things that I know are current challenges. And then at the end of the month, I read through it, and I can see these things that seem to add up.
Right.
It's actually saved me from surgeries. It's helped me to diagnose and tell your doctors what's really wrong.
Yeah.
I mean, it's been phenomenal for me and for my daughter. And I'm so thankful I did it. So I put that in, and that's my parent perspective. So I try to call down a month worth of journaling into one paragraph. Because I understand doctors don't have the time for that. I get that, and I want to appreciate their time. What I'm wondering, though, with that type of a natural history, are there specific things that are really, truly key that the doctors and people would need?
For those specific, for that information? What's going to be the most helpful?
I think that depends on your disorder.
Absolutely.
I mean,
certainly things like quality of life and are important, especially for the rare disease community.
Yeah.
Sorry, I didn't mean to interrupt you.
No, please.
But I think, and that's one of the things that we're working through with NORD is that we have right now our first survey launch has these covers these elements that are sort of all across the board, which I'd be happy to share with you if you want to talk later on. But then it's going to be moving on to what they're calling the disease-specific registry portion, and that's really going to be based on what is the etiology of your disease. But yeah, if you want to talk afterwards, I'll be happy, and I can give you information on what NORD is measuring for natural history information.
I do want to point out, you said something I thought was interesting. You said, this is just a parent perspective.
Right.
What you described is actually basic research, right? So
Absolutely.
you
Awesome.
are the research.
So,
you are the
congrats
person
to
doing
you.
the research. So
Yes.
that's awesome. So
I was going to ask, do you have a scientific advisor that you're working with, or a specific expert?
Have you talked to any other patients that have been affected by your daughter's disease?
Actually,
Or
because of this
parents.
conference last year,
Why, thank you. Because of this conference last year, I actually put on my Facebook page what my daughter's diagnosis was, did a cull down of it because it's pretty lengthy and verbose. And literally within 2 weeks, I had 3 doctors call me, and I had 4 parents contact me from throughout the United States saying, my daughter has very similar presentation, your videos are similar, this is what we think we have, and we've been in contact ever since. And I've been sharing, because I've been using a lot of novel and nootropic supplements and drugs that have actually made far more impact on my daughter's life than any traditional medical therapies have. And I've been sharing those with the doctors and with the researchers that I also contacted because of the information I got from this conference last year.
That's
awesome.
great. So then what I would suggest to you is that you collaborate with those people to help you guys decide
Yeah.
what are the most important aspects of that specific disease, because that's probably going to be where you get the most useful information.
And build that community.
Yeah.
I mean, do whatever you can to build that community. That's going to be your most valuable resource.
Right, absolutely.
Thank you.
more questions? There's a gentleman right here.
Hi,
Oh, sorry, you already have a mic.
I'll run it up there. I just want to tag on that to say that I think instead of PI for principal investigator, we should have parent investigator.
Right,
should be the new
absolutely.
PI.
That's
I
great.
like
I
that.
love it.
So I had a little aha here that I didn't realize, and I apologize if everybody in the room already realized, but you said that Dr. Braverman recruited for the natural history study through clinicaltrials.gov.
Well, she had registered it, through clinicaltrials.gov.
So that's how you can recruit patients into that?
That's certainly one resource that you can look at to find out about the clinical trials that are available is the registry of clinic. And again, clinicaltrials.gov is something that was a patient advocacy organized effort. So I think if you take a look at it, I think they have search. Has anybody?
Searchable.
Sure,
Yeah,
it's searchable by disease.
Yeah.
I mean, it's hard because these diseases are hard to quantify,
Right.
but you can search on there for any of those trials. And there's contact information, certainly for that trial, to who to contact. That's the
Yep,
coordinator.
and they give you just a rundown of what are the goals of the study.
Absolutely.
Yeah, so I know all that part for the clinical trial. We just are starting with one of our scientific advisors, a natural history study. Literally just cut the check and we're about to start. But that can get listed on
Yes.
clinical trials
Absolutely.
and recruit?
Yep.
Absolutely.
Absolutely.
Wow.
Yeah.
Isn't this the greatest conference? Was this coming up here? Am I allowed to hand it off for you?
He's right here, Nicole.
Right.
Hi, I just wanted to share an observation and ask a question at the same time. I just finished developing an autoimmune registry for the American Autoimmune Related Disease Association. One of the things that I came across over the past couple of years that I've been here and traveling to other summits, patient data is the thing right now, which is good. But what I see is lacking is the following of industry standards, if you will, as far as data collection, data normalization, continuity between platforms and designs. So I'm wondering if you're aware of any organizations or any persons that are out there that can help the other patient advocacy groups in developing their platforms, so that what'll end up happening eventually is you'll be data rich, but you'll be information poor because
Right.
you won't be able to share
Exactly.
across platforms.
Right, and I think that's something that, I think that's an ongoing sort of issue that as we continually, more often utilize these registries, that we're gonna have to face on a broader scale. And in fact, and this is something that we have discussed with using the NORD registry platforms, talking about translating this information into another platform if that's something that we need to do. And then also, I remember in that Duchenne Connect registry paper that I read, they came to the conclusion that this is useful information, but there needs to be better standards towards collecting that information, and there needs to be more clearer guidelines towards the collection of that information. And I think as this evolves, this is going to become more and more of an issue, and I'm glad that you brought that up.
Hello, my name is Misak Zetlian. I'm the parent of a child with a rare disease, congenital disorders of glycosylation, PMM2, and father of a child who passed away of unknown reasons, which can be related. What advice do you have for parents and patient advocates when a child in infancy passes away or early, undiagnosed or misdiagnosed or partially diagnosed, on how they can continue and submit tissue samples and blood samples to maybe go backwards in time to diagnose an undiagnosed disease, or properly categorize and provide information for a patient registry, either existing or developing?
That's a really great question. Do you want to?
Yeah, I'll take that. So when my daughter passed away, we actually banked her tissues, a lot of her tissue for research. And I think this group, in particular, should be cognizant of that opportunity. If you have a rare disorder that's terminal in nature, look into that. The platform we use is at the University of Maryland. They have a tissue bank for neurodegenerative diseases. And the time to start thinking about that banking is when your child is still here and when your child is doing okay. And I was fortunate to have that information before my daughter passed away and have time to digest it, I think, and be as prepared as one can be for that eventuality. So I highly recommend patient advocacy groups, parents of rare diseases, to look at the possibility of
It's not an
banking
easy
tissues.
topic to talk about.
No, it's not, and it's very sensitive in nature. You don't want to smash people's hope, but
Right.
there's true research value in these tissues, and I think it's something that needs to be talked about openly, but sensitively with patient groups and parents. And I think in terms of your child that passed away, getting as much data together as you can, and seeing if there's anything that correlates with your child that's still living, and just collaborating with those physicians and scientists, I think, is
Right.
crucial.
And that is natural history data collection.
Absolutely.
Biobanking, I didn't discuss it in any sort of great detail, but that is a part of natural history data collection. I think that's particularly useful for rare disease groups like us.
Yep. Thank you.
One more question, then we'll break for the.
Okay. Hi, I'm Steve Grossman with ADCY5.org. We're a neuromuscular disorder, often gets misdiagnosed as CP, epilepsy, or mitochondrial disease.
Right.
We're ultra rare, so we have about 100 people around the world we know, have kind of a loose, I wouldn't call it registry, maybe 30 people on our list. And actually we've been able to put together a stem cell creation study
Fantastic.
with Rare Science
That's
and CERM.
Yep.
So we've kind of skipped over the natural history study. We have stem cells cooking,
Right.
and now the clinicians got kind of antsy because they saw us getting ahead of them. They want to do a natural history study, and they haven't been the most collaborative of clinicians and researchers. So
Okay.
that's where I'm going to kind of head with this. The patient community has also had data issues with genome. It's identified through genome sequencing only,
Right.
whole genome sequencing. And a lot of us have had our genome sequences withheld from us. We've had other data that was part of other studies and research withheld. That was our data. And one of the things that our patient community is saying, okay, we're going to do this natural history study. What about the data? Are we going to be able to get at least our own personal data back to us during it and after that? And perhaps we want to do that in cooperation with whoever is doing some of the stem cell stuff that's going on. So, patient data
Patient data. So
and patient access. I guess that's. Tell me
Right.
your thoughts.
I was just going to say, in terms of your research community not being as collaborative as you had hoped they would be, do you get together as a research? I mean, do you bring people together? Because we've found that just getting people around the same table and at the same time being able to see the patients there is just crucial.
Yeah, so we're a year old. We've gone from 2 to 100 in a year. And
Okay.
the way we did that is through Movement Disorder Society. Some of the key thought leaders were there. We were able to get about 10 families.
Yep.
And what we did is we did 15 minutes with a panel of all of them. So 10 of them were in the
Right. Oh,
room.
fantastic.
We
That's
trotted
great.
every family through so they got a real good look at it.
Good.
And everyone was all lovey-dovey at the meeting. And then...
So you have to
And then-
continue to foster that.
I know, and it's tough because we only have a few people working with us in the organization, and things drift. And it's like we ask for something, so why do you want that? You're just a parent. You don't know anything. I mean, that's kind of the attitude with a couple
Definitely
Right.
do
people.
your best
people,
to change your
and
attitude.
there are people who are.
You're,
Yeah, I know.
you're
There
a
are other
parent,
people
you know
who are.
everything about
Exactly.
your child.
Yeah.
Exactly.
You don't know nothing. You know
I know.
everything
Yeah,
about
yeah.
your child. So
I think in terms of trying to initiate some sort of natural history efforts, even if you don't have the most collaborative group of researchers, if you can just hone in on one person, that will be enough to at least start the efforts going. And then as it grows, you can start pulling in other people because they will see the value of this as well. And then maybe that's a little idealistic, but I think
No,
that's
we're about to go scorched earth and not give them an endorsement letter. I mean, or at least threaten that. I mean, that's where they are. But on the if patient access to data, that's
Yeah.
something I'd really want to hear about.
yeah,
Or your thoughts.
patient access to their data.
So I think the approach that we've taken with, like we've talked about, we haven't launched the NORD survey yet and collected the information, but we've had discussions as to what we are going to do with this moving forward. And we're very fortunate to have a group of people that do work really well together, and they are actively engaged with us. So our sort of take on it is that we want to be able to share this information
Yeah.
with our clinicians, with our scientific researchers. But I think that being said, it's gonna vary based on the type of vibe that each different patient advocacy group has.
Right.
So I think that's probably a much more personalized sort of decision to make across the different patient advocacy groups. I'm sorry if that's not extremely helpful, but I'm just telling you what our experience
Yeah,
has been.
and I want to kind of piggyback on something Kent said earlier about mining the data. So I don't know whether you're talking about clinical observations and the patient having access to that data from a clinical exam, essentially. Well, I think then you just have to, communicate with that physician and, just share with them the importance of empowering the patients in their home, in their local area to have
Right.
that data, because that's just crucial, I think. And
Absolutely.
And then, in terms of getting that data from other places, our clinical research coordinator for Dr. Braverman, she is the one that seeks out the data from the other centers.
Yeah.
So
and that was,
With consent of
with
the
consent
patient.
from our families to
Yeah.
be able to contact these hospitals where their children's hospital records were.
I mean, I think on the issue of access to data, it is complicated depending on whether it was part of a trial, whether it's a general genetic testing, what the consent forms were. Generally speaking, you should have access to your own data regardless
Absolutely.
of what it is.
Right.
It gets a little complicated if it's in a trial, if they're trying to answer specific questions. It depends, especially in genetic disorders. We're running genetic trials right now where we're very careful about the information we give back to the patients because we don't even understand what the data means yet. And so making incorrect linkages between the data and symptomatology can be challenging as well. So I think the caveat always is, we have to be careful. at the end of the day, I mean, it is your data. I'd have to see what the consent forms were, but when you go into those testing methodologies, those are conversations that have to happen up front.
Absolutely.
It's not as though you're just having a CBC or a chem panel run. You get those very easily. It's a much more challenging discussion around genetic and genotyping and gene sequencing. But in your case, and certainly in the rare disease space, everybody's trying to understand what those things mean. So I think it is a case-by-case basis on how those were run. But generally speaking, patients should have a collaborative access with those physicians that are doing that research to look at that data together and try to make sense of it.
Yep.
I agree.
So-
And I think the more highly sensitive that information is, you can set down, like you said, these sort of ground rules from the get-go within consent forms. If they have access to some data, perhaps it'll be de-identified or something like that, and that might help give information without breaching any sort of privacy or confidentiality.
This has been a fantastic discussion. There's lots more to have in the hallways. It's 11:27. You only have 3 minutes now to get to the next session. But I want to thank everybody.
Thank you.
Thank you,
If
guys.
you have any questions, find everybody in the hallway. We're happy to talk.
Thank you. Thanks, Ken.
Absolutely, guys. Awesome.